coastal water. So this scarcity of water is going to be a big concern. Water availability today is 1,100 cubic meter per capita, which is only 12% of the global average. So most of the constraints are due to climate change and demographic expansion, and evolution of land use is affected. So because of the climate change, we are seeing more and more impacts of drought. And what you see there is the vulnerability of society to droughts. And in our region, the societal vulnerability is dramatically different and is really changing. And you see all those sectors are going to be impacted by drought. Not only agricultural production, but food security, but also energy, transportation, tourism, you name it. All the sectors are going to be tremendously impacted. So I'm not going to go through all the details here on this slide, but you can see that effects of drought on agriculture is going to affect almost every single step in agriculture that you can talk about. So that is why in WMO we organized along with FAO and UNCCD the high-level meeting on national drought policy. Despite the fact that in the world there are for more than 5,000 years we have been facing droughts, of all the countries in the world, only one country has a national drought policy, and that is Australia. We want every single country in the world to develop and implement national drought policy. Why is that needed? Because if you have a drought plan, it depends on which political party is in power. Some parties are more active, others are less active. And then when political parties changes, the plans are not active. But if you have a policy that is a legal responsibility on the part of the government to help the affected communities, and that is why we organized this meeting. And then we try to address that issue as to how we can really reduce the societal vulnerability, and we have developed a number of recommendations as to how we can help the society. And finally, talking about climate change, we need to bring together solutions through integrated assessments. It's not enough that we look at one sector. Climate change is going to affect all sectors in the society. And you need to have an integrated assessment. You start with socioeconomic scenarios, look at agriculture, water, livelihoods, forests, coastal zones, natural ecosystems, livelihoods, and so on. And then through this, we will be able to address the whole issue of climate change. Thank you very much.
Many thanks, Dr. Sivakumar. شكرا جزيلا للدكتور سيفاكومار. أما متحدثتنا الآن فهي أستاذة الطب وأستاذة في قسم علم الأدوية في جامعة مونريال ومديرة مختبر الكيمياء الحيوية للقلب والشرايين في مركز CHUM للبحوث، تهتم في بحوثها بالخلايا الجذعية والمؤثرات فيها مع اهتمام أساسي بالبحوث حول أمراض القلب. متحدثتنا الآن هي الدكتورة جولانتا غاتكوسكا. Dear Dr. Gatkoska, the floor is yours for 15 minutes.
For your Highness, Professor Batiki, Professor Hassan, and ladies and gentlemen, it is really a privilege for me, and I feel very honored to be in your splendid country, which has a legacy, historical and spiritual legacy of Roman, Byzantine, and Islamic culture. On this slide, you see the paintings made by a Canadian painter of the hospital, Hotel Dieu. It has been the first hospital which was organized by French nurses, Germans, on the North American continent. So I would like to entertain you today about, it will be pure science, about the new system which exists in the heart and which is extremely important in the cardiovascular problems which about all we are having. So, I'm sorry, I would like to try change, but yes. So I'd like to go back about 400 years before BC, where Aristotle wrote that heart is a center of thoughts, and the brain contributes to the vital fluid circulation. We know very well that the second thesis of Aristotle has been already proven, but I think that very slowly we are trying also to prove the first one. So, I'm very sorry for this. You know, in the 60s, several groups of researchers in the world had noticed that stimulation, mechanical or chemical stimulation of the certain regions of brain, induces diuresis and natriuresis. And from all these studies, the area of third ventricle appeared, AB3V, appeared the most important. But nobody knew how the brain could affect the kidney function until the discovery of natriuretic peptide in the heart. It is a group of peptides which are very potent diuretic and natriuretic, and it is Canadian discovery of Debold which shows that if you inject in the rats the homogenates of atria, rat atria, the very potent diuresis and natriuresis issue. So we developed the methodology for measurement of natriuretic peptides and found that this peptide exists also in the brain. So the idea was that probably during this mechanical stimulation of brain, there is a release of AMP and diuresis which issue. So we repeated experiment which has been done in 60s. We injected, injected carbachol in the third ventricle and indeed observed the very potent release of AMP, and when median eminence was destroyed, the plasma AMP was completely eliminated. However, the quantity of AMP which has been found in the brain was very small, so it was inconceivable that during the manipulation of the brain, there is such a thing in nucleostratus solitarius, AMP-nergic system in the brain, serotonergic system. We did this by blocking these pathways. And finally, as I mentioned, median eminence has been shown to be very important, and we performed neurohypophysectomy of the animals, and we induced volume expansion, and we noticed that there was, I'm very sorry for this, and we found that there was no release of AMP in neurohypophysectomized animals. In posterior pituitary, there are two hormones, oxytocin and vasopressin. At this moment, we knew already that vasopressin may stimulate the release of AMP, but only by hemodynamic effect. So our candidate was oxytocin, and we injected oxytocin in the, this is what I mentioned before, in hypophysectomized animals and in lobectomized, when only neural rope was eliminated. Blood volume expansion did not induce the AMP. Therefore, oxytocin was our candidate. We injected oxytocin and indeed observed the dose-related release of AMP and diuresis. Therefore, if indeed during volume expansion oxytocin is released from neural node, so goes to the circulation and releases AMP from the heart, therefore we should have oxytocin receptors in the heart. And indeed, we have shown this by the different methodologies, by in situ hybridization, by autoradiography. Oxytocin receptor exists in the 4 chambers of the heart. And I would like to attract your attention to these slides. We wanted to eliminate the neural system, and we performed perfusion, heart perfusion of oxytocin in the presence of oxytocin and antagonist of oxytocin. And as you see, as you see on the left diagram, the antagonist of oxytocin not only eliminated, inhibited AMP release by oxytocin, but also the basal value suggesting that oxytocin is synthesized in the heart. So the whole system of oxytocin, the receptors, and the peptide are present in the heart, and it was the first time demonstration that really heart is contains and synthesizes this hormonal system. And then I will show you what this system is doing in the heart. But it was not the first demonstration that heart is not only the pump, but also endocrine gland, because for the first time the synthesis of this hormone, natriuretic peptide, has been shown. So this is immunocytochemistry, which shows that indeed oxytocin is synthesized in the heart. And what is interesting is that we studied the development of oxytocin in the heart and in the hypothalamus. And as you see, in hypothalamus, the oxytocin synthesis is increased during the development, but in the heart, completely opposite. It is extremely high in the fetal heart and in the newborn heart, contrary to hypothalamus. Therefore, this suggested completely different role of oxytocin in the heart. On this slide, you see what is interesting, that heart contains also the enzyme which the retinoic acid affects the synthesis of oxytocin and oxytocin receptor in the heart. And certainly you know the deficiency of retinoic acid as well as excess of retinoic acid induces malformation of the heart. So retinoic acid affecting the oxytocin system could suggest that the oxytocin system is important in the heart maturation. So we showed also that the same system exists in humans, and in human, in the newborn, is much higher expression of oxytocin receptor than in adult, once more suggesting the possible role in the heart development. So making the short summary, we have shown that oxytocin and oxytocin receptor are present in the heart, and that retinoic acid stimulate the oxytocin system. And this observation led us to make the hypothesis that oxytocin is important during heart maturation and eventually during differentiation of the stem cells. And on the next slide, I would like to show you the results. We used P19 stem cells, which is a very model to study differentiation for cardiomyocytes. And we exposed the P19 cells, and we obtained very shortly, after 2 to 4 days, the beating cardiomyocytes. Unfortunately, you have to play with your imagination because the program did not work. Normally, you should see the beating, contracting cardiomyocytes. So we characterize those cells, and we have shown that those cells which were formed upon oxytocin effect are functional cardiomyocytes which adsorb rhodamine. Rhodamine is adsorbed, is a marker of mitochondria. Those cells need a lot of energy for the beating. So also, what is the role of this system? It was very interesting for us. So as you see, oxytocin decreases contractility and the heart rate, and oxytocin antagonists reverse this effect, showing that oxytocin is playing a role in the slowdown the heart. Furthermore, we have shown that oxytocin also increases the viability of the cells. These properties of oxytocin led us to make the hypothesis that oxytocin is a cardioprotective hormone. The whole system is synthesized in the heart, and the whole system may protect the heart. So we did this in experimental, we tested this hypothesis in experimental model of myocardial infarct, which is a very classical model, ligation of descending artery. We performed several measurements, and since we are, I think, running shortly the time, I would like to show you quickly the results. So the treatment of oxytocin during 3 or 7 days improved the heart work, as it is shown on fractional shortening. It has an effect on the structure of the heart, decreasing hypertrophy. It has also effect on proliferation of the heart and on immune system. Oxytocin decreases the cells, the factors which are important, which induce the immune system. So we found that oxytocin has indeed beneficial effect on the heart by the fact which are stated on these slides. And I will skip this. We looked at the, yes, okay, so I will conclude and I will tell just a few words that oxytocin in the heart is increasing glucose uptake. By this is increasing viability of the cells, decreases apoptosis, affecting immunitary system, and by this fact we consider that it is a very cardioprotective hormone which is synthesized within the cells. And I would like only to go to the maybe last slide, the words which Professor Varna wrote, that recently the heart was considered not only as a thought, but also is a very important hormonal organ, and since oxytocin affects the relations between the people, it is called love hormone. Thank you very much.
Thank you, Professor Gatkoska. شكرا للأستاذة الدكتورة جولانتا غاتكوسكا. والآن أستأذن سيدتي صاحبة السمو الملكية الأميرة سمية بنت الحسن بأن تتفضل بتسليم دروع المؤتمر السادس للبحث العلمي في الأردن. درع مقدم لهيئة المديرين يتسلمه رئيس جامعة الزيتون الأردنية. أنا هذا مكتوب عندي. رئيس هيئة المديرين إذا تفضل. درع لرئيس الجامعة الأستاذ الدكتور رشدي حسن. درع للأستاذ الدكتور أنور البطيخي رئيس الجمعية الأردنية للبحث العلمي. درع للأستاذ الدكتور تركي عبيدات نائب رئيس الجامعة. درع للدكتور أحمد راتب العبوش. دكتور رتنا جوش. دكتور جولانتا كاتكوسكا. دكتور منافا سيفاكومان. دكتور هاكون أرسنال.